An analysis of differential gene expression in hereditary hemorrhagic telangiectasia. HHT is a rare autosomal dominant disorder that causes vascular malformations. Most cases are caused by ENG or ALK1. ENG is found on chromosome 9 and causes HHT1, while ALK1 is found on chromosome 12 and causes HHT2. Mutations alter the transforming growth factor beta signaling pathway. The diagnosis of HHT is difficult because it is a very rare disease. It is an orphan disease which has a lack of awareness, and there is no current treatment for it. This project focuses on the gene expression of HHT1 in both males and females, comparing them as well as with HHT2. We hypothesize that subcombinations within our larger dataset would display differentially expressed genes.
For our methods, we use the NCBI GO2R analysis web tool. There were 3 things that we analyzed. The first was male versus female HHT1, where 16 male samples and 22 female samples were compared, where we admitted the sibling and control data. Next was control samples versus telangiectasia HHT1. We omitted non-telangiectasia and sibling data. We ran an analysis of the combined HHT1 and HHT2 groups. Non-telangiectasia and telangiectasia samples with sibling data were omitted. For all of these, we only collected significant results with a p-value of less than 0.05, and we also ran the accession numbers through NCBI's gene database. For the male versus female HHT1, we found a total of 250 different genes between male versus female. Only 73 of these genes were found to be significant, and of those 73, 6 of them were not sex-linked. We did not consider the sex-linked genes because they were expected to show a significant difference. KDM5D and CD99 both affected the immune system. KDM5D encodes a protein with a short peptide that's a minor histocompatibility antigen that can lead to graft rejection of male donor cells in female recipients. CD99 encodes cell surface glycoprotein, which is involved in several things like leukocyte migration, T-cell adhesion, and T-cell death by caspase-independent pathway.
So the analysis of control samples versus telangiectasia HHT1 returned 26 genes that were found to be differentially expressed. The top 2 most significant genes were CDH19 and CNTN2. Both of these genes code for cell-to-cell adhesion proteins, which are important for blood vessel formation. There were 4 others that were very crucial in regulating angiogenesis and cardiovascular homeostasis. Most interestingly, though, FGF12, which belongs to the FGF family, is not involved in angiogenesis that is known. However, several others in the family are. The results for a combined HHT1 versus HHT2 returned 1 significantly differentially expressed gene, and this was ENG, which corroborates existing literature, and this one is directly involved in the TGF-beta signaling pathway. And interestingly, the ascension code for the variation found in our dataset could be found in AlphaFold, which models protein structures. So the variant found in our dataset is on the left, and the healthy protein structure can be seen on the right.
Overall, we had a few strengths and a few weaknesses in our study. In terms of strengths, we were able to elucidate 12 additional genes that may play roles in development and worsening of HHT. We found that our hypothesis that we would find a significant difference in gene regulation of HHT1 in males versus females was correct, and overall these findings contribute to necessary research on establishing genes associated with the disease. In terms of weaknesses, several significant genes that were found weren't associated with GenBank, and because of this, their function couldn't be explored. Additionally, when we were using the NCBI gene database, the server went offline, so we had to use GeneCard to determine the function of the remaining genes. And also, the datasets for this subject were scarce, making our conclusions potentially incomplete. Lastly, our study has an important broader significance because HHT is a rare disease and therefore doesn't receive a lot of attention in terms of research and funding. Because of this, it's been termed an orphan disease that isn't completely understood. 85% of HHT cases can be linked to well-established genes, but there are still 15% that can't be confirmed. So using available data to assess genes and mutations associated with the disease is important.